Indiana University East, Department of Science, the double helix structure of DNA. So, to understand how this is developed, we first look at some experimental data on the base composition of DNA for a variety of different organisms. So here are different organisms. And this is something that leads to what's known as Chargaff's rules, after the name of the first person who did this. If you look at this, you'll see that while different, if you look at the ratio of adenine and guanine for different organisms, the ratio between adenine and guanine varies. However, you can see that the number of adenine bases is about the same as the number of thymine bases, while the number of guanine bases is pretty similar to the number of cytosine bases. Why is this? This is one of the pieces of evidence that was used to develop the Watson-Crick base pairs, which are put into the Watson-Crick double helix structure. Here you'll find that hydrogen bonds can be formed between guanine and cytosine, as well as between adenine and thymine. And these hydrogen bonds are heavily responsible for the specificity between of base pairing. Now, there are a couple things to note. Number one is kind of mnemonic. If you look here, guanine and cytosine both have curves in the letter, whilst adenine and thymine have straight lines. So those with straight lines base pair with each other, those with curves base pair with each other. Next is the fact that you've got 3 hydrogen bonds between guanine and cytosine, whereas you've got 2 between adenine and thymine. Thirdly, you should note that, well, there's no thymine in RNA. However, uracil can base pair with adenine. Similarly to thymine. So it can take the place of thymine in base pairing with adenine. Now let's have a look at this. If you look at these base pairs, superficially you can see that the GC base pair here looks in overall shape between the guanine cytosine and the adenine thymine base pairs, which means that really what goes in a base pair and the overall shape of the double helix doesn't vary significantly as you go down. And this helix structure, like kind of this circular staircase type structure, is the double helix. Now, a few things you should be aware of. Number one, firstly, so you got the bases in the inside here, and you've got the sugar phosphate backbone going outside. Okay, so you've got 2 polynucleotides. That for the base pairs form your double helix. And the other thing to recognize is that, so you've got these pairs, they're stacked together on each other, and then let's look at this. So here I've got my 5' end here, and then it's the 3' end here. Now the strands are anti-parallel to each other. What do I mean by this? So if you look at this 5' 3' here, the other strand goes the other way around. So it's 5 prime here, going to 3 prime here. So how does that pan out? Well, it means that we have to look at that when we see what sequence base pairs with other sequences. So let's look at this. I have the TATA GTC. So now let's write this. This is my 5 prime n, and this is my 3 prime n. So here, my 5 prime n is going to base pair to this, and 3 prime n is going to be to C base pair G, T has A, G is C, A is T, A, T, A. So therefore, the complementary strand to the original, the TATA GTC, this strand, is the, and I have to look backwards, GACTATA. Okay. Now what holds? So what forces hold the double helix together? Now firstly, you've got the hydrogen bonding in base pairs, which does contribute heavily. But the other thing that's very important, in fact one of the probably more important, is actually base stacking. What we mean by base stacking is this. If you look here, so as you may recall, there's a delocalized pi cloud on each base. And you can see here that on top of each base, the bases overlap on each other. So this the overlapping pi clouds end up having an attractive interaction. And this is what's known as base stacking, which is one of the most important forces that keeps the double helix together.